I'm ready.
All right, let's begin with the final talk this morning. I'd like actually to suggest a slightly different name, maybe 10 women and 1 man, period. But that was not the title, I'm afraid. 10 men and 1 woman.
Okay.
Arndt von Haeseler.
Yeah, thank you very much for giving me the opportunity to present some really preliminary work. It just happened that the numbers were 10 and 1, and I try to explain how we came about these numbers. This is all joint work with Ingo Ebersberger, and I was actually hoping to get a bit further with the analysis of the data while I was staying here, but for one reason or the other I didn't really get to do much on this interesting project. But in any case, what I really want to do with this little presentation, I want to introduce you into the problems one encounters if one studies really a lot of data. But it's not only about reconstructing trees, and in fact this example I will give here, the tree reconstruction problem is not a problem at all, because we are only looking at maximum of 5 species, so you can test all the trees. But it's also something, what is it you do at the end once you have collected a lot of data, and here we really have a lot of data. And then somehow you have to go back to biology and try to start to find an explanation how the variation we observe in a genome really came about. And I will give 2 explanations of that were, one of them was published last year, and the other one is the explanation we give. And then at the end you will see that we have to do more on the modeling side to understand sort of the probabilities of real complex models biologists come up with, or we do come up with. But I think because the title of the conference or the workshop is Future Direction, Phylogenetic Methods and Models, this is a nice talk just to set the scene, what we have to expect in the next future if more and more data are actually arriving and are waiting to be evaluated. And it's, by the way, it's also an interesting topic. I learned a lot from reading a lot of different papers from different fields of science, just to make this number sound plausible and not to offend man or woman. What's it all about? We are, of course, interested in our next relatives, orang, gorilla, chimp, and bonobo. And of course, we all know the big question was for a long time, who is our next relative? And analyzing a lot of data, it is from 5 species, human, chimps, gorilla, orang, and rhesus. Human, chimps, and rhesus are completely sequenced these days, so we have the complete genomes, about 3 billion base pairs. And from gorilla and orang, we have quite a lot of number of shotgun reads, which can be combined to 25,000 multiple sequence alignments. So this is a huge number. It's more than 2.5 million base pairs that you can analyze. And if you concatenate, and it's 25,000 because due to the shotgun reads, you only have short pieces from the genomes of gorilla and orang, and so you get the alignments distributed more or less randomly across the 46 chromosomes of humans. So one can do an analysis of concatenating all these things, and one can do an individual analysis of all the 25,000 alignments. And because we have only 5 species, you can use your favorite method and actually do an exhaustive search and reconstruct the tree. If you do this, you end up with a tree like this, taking the rhesus et al. Group, human, and the 2 chimp groups. Actually, bonobo we don't have in our data set, form a sister group to the exclusion of gorilla and orang. And if you like, you can put some datings on here, which are not really relevant for the ongoing story of this talk. The data that we analyzed are provided by a recent paper in Nature, Genetic Evidence for Complex Speciation of Humans and Chimpanzees by Nick Patterson and co-workers. And this data plus some extra data was also analyzed later by Ingo and Ebersberger and co-workers appeared in Molecular Biology and Evolution this year. We used their data but performed a more classical analysis using standard sort of phylogenetic theory and results from population genetics. And the nice thing is that Ingo Ebersberger gave a presentation of most of these results at the beginning of this phylogenetic workshop in September. And today you will see sort of an ongoing continuation of this work. So this is a phylogenetic tree. However, if you look at the 25,000 alignments, maybe it's a bit, this time we have reduced the alignment, because the number of trees, because we only wanted to have trees that receive a high support. So what you see from the possible trees we have, we do not always reconstruct for each alignment the precise speciation tree. And of course everybody in the field knows that there is a fine distinction between the species tree and the gene tree. And if you have two speciation events that are following closely in time, then it's very likely that due to incomplete lineage sorting, you may have a gene tree that is deviating from the species tree. In the case of the human, chimp, gorilla, for speciation events, one sees that the most frequent gene tree is the tree that groups human and chimp with gorilla. And we have two other trees that occur at roughly the same percentage of 11.5%, where the chimp is sister to the gorilla, or where the gorilla is sister to the human. And this is exactly what you would predict from population genetics theory in the context of speciation event, if you look at a rooted three-species tree. And the reason for this is pretty simple. If we start looking at sequences from different species and reconstruct that tree, we tend to ignore that a species tree actually has a volume. And the gene is transmitted from one part of the chromosome to its ancestor in sort of this discrete way here. So we can trace back the history of one gene through a series of really existing ancestors, and eventually the two lineages here from human and chimp will merge, and then the trace back continues, and the lineage from gorilla in this case and human chimp ancestral population will also merge. And then you have this green gene tree that agrees with the speciation event. And of course, what is necessary is that during the existence time of the most recent common ancestor species of human and chimp, these two lineages need to merge. If this lineage is short, or if this time span is short, then the probability that these two lineages will merge gets smaller, and we will have a higher chance that all three lineages will sort of arrive at the most recent common ancestor of all three species. And then standard coalescence theory in its sort of simplest form applies, then each tree topology is equally likely. Of course, this branch is never zero, so there is a small chance that the tree that groups human and chimp will occur at a higher frequency. And that's exactly what we observe in our data set. We also can make a prediction. If nothing is really wrong, then the two alternative trees should occur at the same frequencies, which they actually do. This is the easier case for a rooted three-species tree, and I guess on Friday you will hear more about the complications that arise when we are looking at 4 species or more, because then you can actually get in real big trouble, which we don't do at this point, fortunately. We have trouble enough. So, summarizing this in one big picture is we have the human, chimp, gorilla. We have several independently evolving genes that where we can trace back the history, and the outcome from theory is basically that we would expect 3 different trees. The red one, which is congruent with the species tree, the green one, which groups chimp and gorilla to the exclusion of human, and the blue one where human and gorilla are grouped together. Note that due to the different tree topologies, it's really difficult to date the exact speciation event here. That very much depends sort of on the data you are looking at. There are also some other issues involved. You have to make sure that these branches are actually supported. Most of the data we had to throw away because we had not enough information to actually support these splits. Ingo explained at the beginning a lot of nice biological implication one can simply get from this picture and the frequency of congruent and incongruent trees. And I will continue with this. So basically the probability that a gene tree is congruent to the species tree is nothing like this simple formula. It's 1 minus 2/3 times e to the minus delta t divided by the effective population size of this population here times generation time. So this is just a scaling factor. We don't really have to care about it. The important thing is that the population time enters as one parameter, and that the time of existence of the ancestral species enters as the other parameter. So a big population size also makes things more unlikely. Formula is pretty old, 1987, and biologists tend to forget the process of finding sort of common ancestors in populations when they discuss phylogenies. And so if you are not aware of this effect, you may as well argue, well, I got this tree with high confidence, so this must be the true phylogeny. And in fact, we can only tell something about the true phylogeny if we have a large sample that gives a solid estimate of the number of the frequency of the trees in the statistical sense. So just to give you one example that the frequency of congruent and incongruent trees is by and large independent of the region of the genome you are looking at. I said we have about 77% congruent trees if we look at the entire data set. If we look at genes, then this number increases a little bit to 79%. And if we look at exons, the number is also something like 79%. So even if we would go into coding regions and in exons, the numbers do not change dramatically. they increase a bit. That could be attributed to selection on certain genes, but we didn't really analyze this in more detail. And again, also the percentage of incongruent trees remain more or less the same. If you look at chromosomes, you also see now significant pattern of clustering of congruent trees or incongruent trees. Here I must admit this picture, is a bit misleading because the sampling is in fact much more sparse. It's just the dimension of the picture here that makes it look much denser as it really is. So long gaps in between. And there are other methods published in PLOS biology, I believe, where people develop a hidden Markov model to sort of walk along the chromosome and model the switches between trees. One thing, of course, that sticks out, and that's the story of today, is if we look at the fraction of incongruent trees along the chromosomes, then we see that the fraction of the incongruent trees of the X chromosome is only 9% or 10%. Much smaller than anything else we observe on the autosomes. In fact, if we then would compute the effective population size using Ney's formula, this number still is much smaller. And even if you would correct for the reduced effective population size of the X chromosome, this number would still stay significantly smaller. So there is something strange going on the X chromosome. And this was also recognized by Patterson and co-workers. And they tried to provide an explanation why we observe the reduced variability. Actually, they talk about variability. We talk about number of incongruent trees, which more or less is the same. How to explain this incongruencies or the reduced variability on the X chromosome. And what they proposed is a rather strange model. Basically, the model says we have an initial speciation of whatever, human and something else. And after this speciation, we had some hybridization going on that then led to humans and chimpanzees. And this, of course, something like this always hits the newspapers because it's somehow good. I don't have to say, so I can stay. I think one projects it into nowadays somehow. We suggest, to quote them, we suggest a provocative explanation for multiple feature of these data, that the hominin and chimpanzee lineages initially separated, but then exchanged genes before finally separating less than 6.3 million years ago. And then one has to invoke a second process, and just not to sort of get the wording wrong due to my not so fluent English, I just copied the page. The low divergency of humans and chimps on the X chromosome. An empirically observed pattern documented in Drosophila, flies, mosquitoes, butterflies, so invertebrates, and guinea pigs, is that the genes having the greatest effect on hybrid sterility and in viability are X-linked. And then there is Halden rule that says if you have hybridization between different populations that have sort of differentiated for some time, then the hetero chromosome carrier, so the males in humans, is strongly affected by selection if we have the inviability on the X chromosome, which makes sense because humans only have one X chromosome and females have two. So for this reason, the second rule of speciation, not fully understood, are thought to be related to Held's rule about hybrid sterility affecting the heterogametic, that was a word I couldn't remember, sex more than the homogametic sex. So male are heterogametic and female are homogametic. And then they suggest a corollary, not that the gene flow between two diverged populations occurs. Chromosome X should be subject to strong and rapid selection to eliminate alleles from one parental population or the other. That contributes to reduced fitness. The presence of multiple hybrids in compatibility logic could lead to selection across much or all of chromosome X as they observe in their data. Well, reduced variability is typically a sign of selection. This construction is extremely complicated and makes a lot of assumptions. Then they have to say something about infertile males. This is basically consistent with Held's rule. And then a viable population could only have arisen if fertile females mated back to one of the ancestral population, for example, the chimp ancestors. Then we have fertile males, hybrids, when producing fertile male hybrids when they transmitted X chromosomes that were almost derived from the ancestral population, so the one you saw on top. And this could explain why humans and chimps are most closely related throughout chromosome X. In wild mice, they observe sort of a sharp transition for the X chromosome and a much smoother transition or gradient for the autosomes. So this indicates that such effects may actually occur, that hybrids may have difficulties in carrying X chromosomes from multiple ancestral populations, and which is, of course, consistent with their model. So hybridization of sort of ancestors of humans and chimpanzees occurred. Strong selection on the X chromosome, and this explains why the variation on the X chromosome between human and chimpanzees is much more. This also explains some other things where Ingo and I think we have an easier explanation for this, but this is the model. Well, we were thinking, isn't there a simpler model? And what is it we then did? Well, I showed you this formula from Ney. In fact, if you use the formula as I showed it at the beginning, you simply assume a 1-to-1 sex ratio. Now we have data for the X chromosome, which has a smaller effective population size, and for the autosomes, and as you have seen on the previous transparency, the frequency of the autosomes of incongruent tree is not really substantially different from each other. So we can use these two equations and just solve for the number of effective females and effective males. This is only sort of 2 parameters, which is well known in the coalescent framework. And we can plug in numbers here. We don't really have to, but for the sake of sort of illustration, we use the 2.1 million here and a generation time of 20 years. It's cancelled actually. For the ratio, it doesn't matter. These were the data. And then we end up with an effective population size of 45,600 males and 4,700 females, which is roughly 10-to-1. Yeah, that's great. So most of you show the same reaction I had. Then I thought, well, the mathematics cannot be wrong. That's the nice thing about mathematics. Maybe it doesn't apply to the real world, but the result is exact. And so I went to Ingo and said, well, it's 10-to-1. And he said, I cannot believe it. This is implausible. And actually, if you read the Patterson et al. Paper, they have a 50-page supplemental. Somewhere they also discuss sex ratio, and they start with a sex ratio of 4-to-1, then they do some calculations which I didn't really bother to understand. And at the end, they say seems so unplausible, so we reject this hypothesis. This is not a rejection in a statistical sense, it's just a hypothesis, a rejection in sort of, not daring to think the unthinkable. Well, so then I thought, well, so what can I do? And this is really something which I think is interdisciplinary work, to find evidence for this. First of all, you ask the following question: is there any advantage to have this extreme sex ratio? So what you do is you consult sociobiologists. They have had a lot of thoughts and actually also a lot of mathematical models. And they come up with sort of reasons which are briefly summarized here. Multiple partners provide female with increased resources such as gifts or food or protection. Females seek out males that will provide superior DNA for their offspring. Female promiscuity is a tool of natural selection for enhancing the gene pool. So it favors not only the survival of the individual female, but in a sense also the survival of the whole population, especially if resources are rare. If you have a lot of resources, then of course you somehow don't have to care about bringing up your child because everything is there. So this was sort of a social biologist's explanation why these things may be advantages. Of course, they do not really specify when they are advantages and what are sort of the true conditions. Well, then I went to anthropology. And actually, if you look up reports from anthropology, you see that polyandry, so that is that a female forms a sexual union with more than one male, actually took place in such remote places like Tibet, in some Canadian Arctic regions, Nepal, I don't know where this is, I try to find it with in Sri Lanka, some areas of China and the Mongolia, somewhere in sub-Saharan Africa, and also in some American indigenous communities. So it's, I will not say it's frequent, but it's occurring in humans. And actually the first known inhabitants of the Canary Islands also practiced it until they disappeared. And so much for the advantage. So there is at least some scattered reports in anthropology literature that these things happen. Behavior. Wow. Chimps. This is from a paper by Doros et co-worker in Nature. Actually, the table is from a book which I couldn't find. So they basically have a list of primates and the chimpanzees, so our next relative, actually have a mean number of male partners per ovulatory period of 8. Close to 10, I must say. And then it decreases, and actually the table explicitly says that in human things are changing so quickly that 1 to 2 is just an estimate. And it gets better actually. Doris et al. Actually used the table to show that the degree of polyandry can actually also impact the dynamics of molecular evolution. And they studied the evolution of SEMG2, the gene encoding ximenoglelin 2, the structural component of the Zeaman golung. And that is accelerated in polyandrous primates relative to its monandrous counterparts. And that's what they show on the tree here, where you have a high non-synonymous to synonymous substitution rate here and somewhere else on the tree. And the chimpanzee actually sticks out with a high selection coefficient of 2.52. And the idea is that if you have promiscuous females, there is more selection pressure on the male semen to be more competitive against all these other competitors, just to block the entrance of the next sperm. So this is a molecular biology indication that this is probably not far off. And finally, I found this nice headed, and this is a narration, is an oral tradition relating the words and deeds of Prophet Muhammad. I found it on Wikipedia. I would have loved to see it on a book. There were 4 types of marriage during the ancient Arab period. One type of marriage was that a group of less than 10 men would assemble and enter upon a woman, and all of them would have sexual relation with her. If she became pregnant and delivered a child, and some days had passed after her delivery, she would send for all of them, and none of them could refuse to come. And when they all gathered before her, she would say to them, "You all know what you have done, and now I have given birth to a child. So it is your child, oh so-and-so," naming whoever she liked, and her child would follow him, and he could not refuse to take it. So this is, of course, circumstantial evidence. The molecular biology evidence and the other evidences are probably not way off, but unfortunately, I must say, this is not the end of the story. Obvious issues are, does extreme sex ratio also explain other aspects of the data? That's something we didn't really look into. 10-to-1 is simple a point estimate. We need a more detailed statistical analysis, and I can tell you that the variability of this point estimate is really big. And I mean big. That's basically following the argument we had early this morning. If you have these stochastic processes, you have a high variability anyway, and now you are computing a ratio, which is probably the worst thing to do, but in a sense the only thing we can do here. So if it's later 8-to-1 or 5-to-1, I don't really care. And then something which I really want to do, what is it we as a modeler or you as a mathematician can do to compare such sort of wild hypotheses to much simpler ones? Because this is a real challenge. This is very much along the line of the talk this morning. One sees speciation in a tree or rapid divergences, and one is sort of misled by the pictures one sees. And if one then does the real statistic, then all of a sudden everything disappears, and the biologists are very, very disappointed and not happy. So here's really something in theory we need to do. And with this, I would like to thank you, and I'm ready to answer questions.
Diagram of the relationships between human, chimp, and gorilla, you had huge sections which would just favor human and chimp relative to gorilla, and then there'd be a switch, and there'd be a big block, a syntenic block, which seemed to have something different. Oh, okay, that's where it appears.
We tested this. You can't see anything.
Oh, okay.
That's again the human eye.
Okay.
Is there any fossil evidence for this?
No. It's hard to find. No, definitely not.
It's not enough to just have sex. You also need to kill the rest of the women.
That's a very good remark. This is actually a problem. What you actually have to have, you have to have a large fraction of women in a population that do never get a child. But on the other hand, it's a difficult issue. I haven't fully understood the relation sort of between things that are happening in a physical population and the effective population size. Because it's not one-to-one, it's only one-to-one in this very simple Wright-Fisher setting. And that makes things really difficult to sort of transfer to the real world. It's just a parameter.
So, one of the things that's been detected in Icelandic pedigrees is that there's a greater correlation of reproductive success from generation to generation in women than in men, which would tend to reduce the effective population size of women compared to men.
I want the reference.
I can give it to you. It's 2003.
Yes. Actually, I would like provocate you.
Oh, great.
Because when we observe what happens in nature, typically it's exactly the inverse. There is one alpha male with an harem of many female. And I mean, I don't have any other, why this cannot happen in the past. I mean,
Yes, but the interesting thing is, if you again look up literature, and it's actually that's where the new DNA sort of molecular biology techniques also changed the view in behavioral biology. If you look up the literature, the alpha male appears to be the alpha male, but all the other males somehow sneak in. And,
Or the women sneak out.
and the women actually sneak out because of the fact that so it's not as simple as it appears on the surface. So all these microsatellite studies have indicated that even in fishes, that the alpha fish does not produce all its offspring. Also the beta, gamma, and however you call these male fishes down the hierarchy are actually contributing to the gene pool. And the females actually actively sneak out because they think one alpha is enough, probably.
and also one other thing, why mathematically it is more reasonable this model and not another model in which you admit that there is a very high selective pressure on the chromosome X, so that the reduction of mutation of the two species is
I tell you why. Because I think people always call up selection if they don't know better. And we have sort of, in these models, we have male and female effective population size. And this is one parameter one can adjust. And that's actually the last remark on my slide. What we really need to do as theoreticians, we need to be able to compare these different hypotheses one way or the other. This one has one parameter. The complex hybridization model has, I don't know how many parameters.
So maybe a related question. So it seems like much of the evidence that you gave has to do with different behaviors in males and females, and it's not clear to me that there isn't a way to incorporate that into the coalescence model. So the coalescence model that you're using is actually symmetric, right? The way that the male and female are represented is completely symmetric, right? So it's not clear that, you know, if you incorporate something asymmetric, you will actually get a population ratio of 10 to 1. It could be that just the asymmetry in the definition of the model will actually lead to a ratio of 1 to 1, right? Or 2 to 1, or whatever.
I don't know how to do it at the moment. That's my biggest problem. So sort of the equations are fine, but I actually don't really know how to do it. And that relates to your question. So on face value, one would assume, well, the birth rate ratio, sex ratio is 1 to 1. So if only very little females actually have offspring, then there must be high female mortality.
There could be larger variance in the number of children.
It
for
could be
male
also
and for
so-
female.
yeah,
There
yeah.
may be other aspects.
Yeah.
Play
So
a role here.
male on average gets 1, females 10. The ones that really contribute. So this is difficult, and that's of course a problem of the coalescence. Everything you see, or most of the things you see, assumes variance 1, I guess.
Stephen?
One source of higher female mortality might be the difficulty of childbirth. Humans seem to have a much more dangerous process of childbirth than many other species. And so if there's a higher mortality in childbirth, then relatively few women who reach pubescence actually might survive with offspring.
That occurred to me too, but I don't know how it is in chimpanzees, for example. In humans, we would all agree, but it's...
Hasn't that changed since the report?
Yes, but that's the time scale. That's, yeah, that's 200 years or 150 years. Yes, that's also something that springs to mind, at least, at one explanation, yes.
A few weeks ago, I was looking at some syntenic maps between different mammalian species, and I can't clearly remember what I was seeing, but I seem to remember that the X chromosome was much more intact across these different species. So it would suggest, I suggested here, that there's greater selective pressure.
Yeah, it is certainly, it is the greatest. There is definitely more selective pressure on the X chromosome. And it's again something which may be simply reflected in this ratio.
All right, I think we should wrap it up here and thank Arndt and all the speakers.